Welcome back, friends. Welcome to another video tutorial from Shomu's Biology. And in this series of videos, we are going to talk about carbohydrate metabolism. And especially, we want to talk about the cellular respiration. So we will look at the different stages of cellular respiration and what they are made up with. So if you think of cellular respiration of carbohydrate metabolism, it starts with a process known as glycolysis. And then the process goes like the modification of pyruvate into acetyl-CoA, that is done by pyruvate dehydrogenase complex. Then the third stage of the process is known as the Krebs cycle, and the fourth stage of the process is known as the electron transport chain to generate energy. So now if we look at the overview of complete process of cellular respiration, it always begins with glycolysis. In this first video, we want to talk about the first stage of this cellular respiration, that is glycolysis. The idea of cellular respiration is simple, that we eat food and the part and fragments of that food after the digestion reaches to our cell. Now inside the cell, all those food particles are broken down into the macromolecules, that is carbohydrate, lipid, proteins. These are the three major macromolecules that they produced after the food digestion, and a little bit of nucleic acids as well. Now what we do is all those fragments that we're talking about, that is carbohydrates, lipids and proteins, all these materials are then taken inside the cell and start breaking down. They start broken down and produce energy, because these molecules contain energy because they are produced by the plants or producers with the help of a process known as photosynthesis. So they produce this food, we take this food, we break them down, and then we utilize the energy. So the idea of cellular respiration is to utilize all those macromolecules to produce energy inside the cell. And to make that, they have different process of metabolic pathways, numerous metabolic pathways for carbohydrates, lipids and proteins. They have separate metabolic pathways for the generation of energy from all these macromolecules. Now here we are talking about the process of carbohydrate metabolism especially. And the carbohydrate metabolism, if you see, what happens is in carbohydrate metabolism, it starts with glycolysis. So glycolysis is a process where we usually take carbohydrate, the simple form of carbohydrate. Let's take glucose, and we convert it into pyruvate. Then the second stage of the process, if we go, then we take this pyruvate, which is so let's take this pyruvate as a substrate and we convert it into acetyl-CoA. Okay, and this step is known as and catalyzed by the pyruvate dehydrogenase complex. Then comes the third part or the third step of this respiration steps, and that is Krebs cycle. And what happens in Krebs cycle? Again, the product of the early state, which is acetyl-CoA, is taken as a substrate. So you take acetyl-CoA as a substrate and they generate and they oxidize acetyl-CoA and all the materials that is produced there at the end of the glycolysis stages and processes. They oxidize it and they produce a lot of energy containing compounds like ATP, NADH, FADH2. Now what happens? All these stages are sequential, starting from glycolysis, then pyruvate dehydrogenase complex and Krebs cycle. After that, after the Krebs cycle is done, then what they take all those molecules like ATP, NADH, FADH2, they take all these things and they utilize the energy stored in ATP, NADH and FADH2 to produce energy currency of the cell, that is ATP. Because whenever ATP is directly produced, it can be utilized then and there. But molecules like NADH, FADH2, they are electron carriers. So there's a tendency in our body using electron carriers as a donor of electrons and driving a set of processes to generate more molecules of ATP. And that concludes the fourth stage of the cellular respiration, and this fourth stage is electron transport system or electron transport chain. And at the end of this electron transport chain, what happens? All these molecules that are produced, they are utilized to produce more and more energy currency, energy molecule, that is ATP. So this is the overview of cellular respiration in our body. If you look, all these stages. Now here in the sequential stages of series of videos, we are going to talk about all these fragments, about glycolysis, about the conversion of pyruvate into acetyl-CoA, about Krebs cycle, and about electron transport system. And we will see their advantages and what they are required for, their processes, and all the detailed steps of each and every process that we've described till now. So stay tuned and watch the video throughout, because we are going to talk about all of them in details. Glycolysis is a process by which the body converts glucose into pyruvate. Now pyruvate is then either converted to lactate or lactic acid, which is an anaerobic process, or it may also be converted to acetyl-CoA for use of that acetyl-CoA in another cycle known as citric acid cycle, that ultimately leads to further conversion of that pyruvate, and then finally through electron transport system it generates energy. Let's move. Now the process of glycolysis can be divided into three different parts. The first part is known as the priming part, where a priming step involves in the requirement of 2 ATP molecules. The sugar splitting state is the second step of the glycolysis. In this step, one molecule of glucose becomes 2 3-carbon molecule compounds. As you can see here, before the splitting we have fructose 1,6-bisphosphate. In the splitting step, that fructose 1,6-bisphosphate, which is a 6-carbon molecule, is divided into 2 3-carbon molecule, namely glyceride dihydrophosphate and dihydroxyacetone phosphate. After that, the third step, or it's known as the energy extraction step. In this step, the energy of the sugar is harvested in the form of ATP and NADH. Both of these molecules, NADH and ATP, are energetic molecules, and 2 molecules of those ATP and 1 molecule of NADH are generated from one of those 3-carbon molecule that was generated earlier. So the first step of priming consumes 2 ATP, while the energy extraction steps provide us 2 ATP and NADH. Now let's look at each of the process in much more detail. In biologically irreversible step is this first step, and in this first step the glucose is phosphorylated into glucose 6-phosphate, and this is the most important part of this step because it is irreversible in nature, and as it is irreversible in nature, the commitment for this step dictates the commitment for the cell to continue the glycolysis process. So this is a rate determining step. Now the enzyme for this step is mainly glucokinase. Glucokinase can be present in liver, and when the glucokinase enzyme is present in different parts of the body, it's known as the hexokinase. The liver is able to regulate the blood sugar level because of differences in the Km values between this glucokinase and hexokinase in the different regions of our body. Okay, and the uptake and regulation of this glucose also mediated by different glucose receptors that are present in different regions of our body cells. Now if you look at this process in details, it comes simply addition of a phosphate group to the sugar or to the glucose from the beginning, and once it's added, magnesium is required for this enzyme to function properly. So magnesium acts as a cofactor, and the most important point that this is irreversible step and this is an energy consuming step, and this is a commitment step for glycolysis. So if this step is done, then obviously the cell is undergoing the rest of the stages of glycolysis as well. So there is no going back after this. The second step of glycolysis, it is reversible step, and it sets up the molecule for cleavage between the 3 carbon and the 4. So if you look at here, this is earlier, if we talk about the structure is modified from glucose 6-phosphate to fructose 6-phosphate, and the enzyme involved is known as phosphoglucose isomerase, which changes the structure of the glucose 6-phosphate into fructose. So it changes the structure into an isomeric form. That's why it's known as isomerase enzyme. Okay, and this conversion is actually a conversion from aldose sugar to a ketose sugar. If you look at here, glucose 6-phosphate is an aldol sugar that is converted to ketose, which is fructose 6-phosphate. Now the difference between aldose and ketose you can look at here. The form is, in aldose this is a hexameric ring, in ketose is a pentameric ring. Now the third step of the reaction is also irreversible step, and in this case it uses phosphofructokinase enzyme or PFK as a process of conversion of fructose 6-phosphate into fructose 1,6-bisphosphate. And this is another step when there is a phosphate group transferred from ATP to the fructose 6-phosphate to convert it into fructose 1,6-bisphosphate. Okay, so 2 phosphates group will be added, and again for the addition it is another energy consuming step, and they also require a cofactor magnesium. Now the key point is this step is irreversible, it is also a committed step and a major point of metabolic control, and there is more than one type of phosphofructokinase inside our body, which also makes it a little bit different in the different parts of the phosphofructokinase activities. We'll talk about the phosphofructokinase activities later. The fourth step is the splitting of the fructose, and this is also energetically unfavorable, but this process is driven, and this is driven forward by the low concentration of the products. If the product concentration is little high, then this process will be blocked, or this step will be blocked. Okay, because as the products that are produced further continues to be removed by conversion, that lower product presence helps them to properly convert fructose 1,6-bisphosphate into the 2 3 carbon molecules. So normally fructose 1,6-bisphosphate is also having 6 carbon. They have 2 different phosphate groups attached. Now it is degraded, it's broken, and it produces 2 different 3 carbon compound instead, and the 3 carbon compounds are glyceraldehyde 3-phosphate and dihydroxyacetone phosphate, and the enzyme associated for this process is known as aldolase enzyme. Now the key point for this reaction, that this is a very important step that intersects with the fructose metabolism, with the pentose phosphate pathway, and the lipid metabolism as well. So this is a very interesting branch point of the process of glycolysis, and this is also a splitting step from 6 carbon to 2 3 carbon molecules. Now if we go to the next step, that is a conversion or interconversion between the glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. Glyceraldehyde 3-phosphate can be readily converted to dihydroxyacetone phosphate, it's known as DHAP, and this is also a reversible reaction, so that they can easily change between their structures inside. The change is very easy to construct depending upon the structures. If you look here, it's easy to achieve. Now the enzyme involved with this process is known as Triosephosphate Isomerase, and the key points to remember is that this step is effectively doubles all the products that are shown here. So once this glyceraldehyde 3-phosphate is a 3 carbon, so whatever thing we'll see downstream of it will be produced from a 3 carbon molecule, but we know actual molecule was 6 carbon, so every of these processes that we are going to see further will be applicable for each of those 3 carbon intermediates, glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. The next step of this reaction is the oxidation, and that uses electron acceptor. That is very, very important in the biochemistry, and the role of electron acceptors, which is NAD+. And this is the step of converting glyceraldehyde 3-phosphate into 1,3-bisphosphoglycerate. And in this case of this transfer, they transfer this energy or electron to NAD. Once it's transferred the electron to NAD, and it also requires the amount of phosphate, because the phosphate will be added there, because we are converting glyceraldehyde 3-phosphate into 1,3-bisphosphoglycerate, because the phosphate group is added to both 1 and 3 carbon positions there, and the electron is transferred to NAD to convert it into NADH plus H plus, which is the reduced form of NAD. Now in this case, the enzyme required for this process is glyceraldehyde 3-phosphate dehydrogenase. Normally this nicotinamide and flavine enzymes frequently shuttle electrons between the metabolic process like glycolysis and the mitochondrial electron transport chain. However, to be used in that way, it must first be transported into the mitochondrial matrix. So whatever electrons generated, this NADH transfers them to the mitochondrial matrix, and then they use that for the process of electron transport chain, which we will also study later. Now the later step is the conversion of 1,3-bisphosphoglycerate into 3-phosphoglycerate. Now most ATP is produced by the electron transport chain. In comparison though, the kinase reaction is an example of what is known as substrate level ATP production. Okay, so what happens in this case, as we know that 1,3-bisphosphoglycerate has had 2 different phosphate groups. Now one of that phosphate group is cleaved and attached to the ADP. Now ADP, once attached to the phosphate group, converted itself into ATP. By this fashion, the substrate is helping to produce ATP, known as substrate level phosphorylation. Now unlike the other kinase in the glycolysis, this is an example of readily reversible kinase activity, and this activity helps to generate ATP for the first time in the process of glycolysis. The enzyme involved is phosphoglycerate kinase, and the cofactor required in this process, like any of the kinase enzymes, is also magnesium. Let's move to the next step. In this particular step, this 3-phosphoglycerate is now converted to 2-phosphoglycerate. It's simply the rearrangement of the phosphate group from the carbon number 3 to 2. Now 2,3-bisphosphoglycerate is an important mediator of this phosphate transfer. It is formed from 1,3-bisphosphoglycerate, and is also important in affecting the oxygen affinity in the hemoglobin in the blood, which we will also see in a separate video. So in this case, the enzyme involved for this process, phosphoglyceromutase, that simply interchange the structure, and the cofactor required, as we told, 2,3-bisphosphoglycerate. Okay, now once 2-phosphoglycerate is produced, now that 2-phosphoglycerate can also be modified in the later stages, and the 2-phosphoglycerate is modified into phosphoenol pyruvate, or PEP. And in this case, this is the 2-phosphoglycerate. This phosphate group remains it is, and water molecule released. Once the hydrogen and hydroxyl released from the third carbon, and the hydrogen is released from the bond between the second carbon, that releases water out of it. So dehydration step occurs, which converts 2-phosphoglycerate into phosphoenol pyruvate. And you know, phosphoenol pyruvate is a very, very energetic molecule. It contains the phosphate and very less those, it does not even have those hydrogen or hydroxyl content as the water is released. So this is a very energetic molecule. The enzyme involved in this process is known as enolase, and this process is inhibited by fluorides. So fluoride are toxic, and those any types of fluorides are actually toxic at this specific region, or they block this specific part of the reaction of glycolysis. The later stage, or the last step of the glycolysis, is the use of enzyme known as pyruvate kinase, which is a very large like an enzyme, and this pyruvate kinase converts phosphoenol pyruvate into pyruvate. And as I told you, phosphoenol pyruvate is a very energetic molecule, and it also contains a phosphate group. So while conversion from PEP to pyruvate, they transfers this phosphate to ADP that makes ATP. So we get the second round of ATP production in the glycolysis pathway. Now there is a very large negative free energy associated with this reaction. It is biologically irreversible reaction, and it pulls entire pathway towards the completion. So this is one of the most vital stages of glycolysis of all. Now this step is also inhibited by different molecules. It is inhibited by the presence of ATP, because presence of ATP means presence of energy inside the cell. If there is a lot of energy inside the cell, cell will not want to produce more after glycolysis or Krebs cycle. So ATP prevents this step, so as alanine amino acid. The liver also controls the step by covalently inhibiting the enzymes by phosphorylating this pyruvate kinase enzyme. If you phosphorylate this enzyme, it gets inactivated. So by this fashion, the most important points of the step: it is irreversible, liver inhibits glycolysis with more sensitivity than other tissues or other regions of our body. So this, in a sense, is the end. Now, once the pyruvate is being made, it is a product of glycolysis, but there are some fate of the pyruvate. Now the fate determined by the presence of air or the absence of air. Now under aerobic conditions, it is mostly converted to acetyl-CoA, okay, which will be shuffled between oxaloacetate. And however, in anaerobic condition, it must be converted to lactate. And once it's converted to lactate, it utilizes NADH, oxidize NADH to NAD+. And in that case, they produce lactate, and lactate can be, and production of lactate from pyruvate can also be used to regenerate NAD+. Now this is especially important in cells that rely exclusively on glycolysis for energy, like red blood cells. In that case, these are the possibilities. And the process of conversion of pyruvate into lactate is known as a lactic acid fermentation, and the enzymes required for this is lactate dehydrogenase.